Okay, we'll pick up where we left off. I'm sorry about that. I fixed all of them so we won't have that problem again. But the outer portion of either whether bacteria or archaea are designed for protection from environments, from changes, and also basically you have this idea of an endospore where the bacterium is able to replicate its DNA, remove all water, and actually encapsulate itself in a hard outer structure so that that DNA lives in parentheses. It's not carrying on any metabolic activity. It's not moving. It's not changing it at all. It's just waiting for an environment where it can do that. That we call that an endospore. And we'll look at an endospore in the next slide or two. But this outer cell wall maintains the cell environment, so protects it from anything. It protects. It's a physical barrier, and basically a pretty substantial physical barrier. It protects the cell if it's put in an environment that is hypotonic or hypertonic. If you remember, we talked about that. That has to do with osmosis and has to do with water moving in or out of the cell in order to equalize the solutes. And so if there's a vast change in water, the living cell can either shrink or it can burst. And this cell wall protects it from that. With bacteria, so one of the differences between bacteria and archaea is the contents of the cell wall. Bacteria cell wall contains what's known as peptidoglycan. That is a network of sugars and cross-linked by polypeptides, just for your information. And the archaea have a liposaccharide on the outside. They do have some of this glycan layer, but the outer is a lipid saccharide, so a fat sugar, I guess you might say in layman's terms. And this is how we stain them. And using a technique that was developed in the 19th century, so that long ago, by a doctor, a physician whose last name happens to be Gram. That's where we get this gram-positive, gram-negative concept. And you'll do this in lab, which will be kind of interesting eventually, where what you will do is you will stain the specimen with violet and iodine, and then rinse, and then stain it with an something that has kind of an orange color. And the gram positive will, because of the outer peptidoglycan layer, will actually absorb that violet color, whereas the gram negative won't. So you'll have a deep purple to kind of a pinky orange mix between the gram positive and gram negative. And this was one of the first advances with regard to medicine, because they could start identifying what bacterium is actually infecting or affecting, I guess affecting by infecting whatever patient they were working on, whether that be a human patient or an animal patient. So this concept of gram positive, gram negative had certainly technological and medical advances to it, but it's done just based on the difference between these cell walls, between the two types of major domains. Archaea, as I said, contain polysaccharides and proteins, but lack that peptidoglycan. Polysaccharides also, not just in archaea, but can form a protein layer called a capsule. This is this concept of an endospore. And that capsule is fairly indestructible. Certainly can be destroyed, but fairly indestructible. This endospore that's created is created with the capsules can provide a sticky layer that can adhere to your throat, by the way, unfortunately, can adhere to something, can also cause these bacterium to stick to themselves. But the capsule can also form the endospore. Well, the capsule encapsulate the endospore. So if in fact this bacterium is suddenly put into conditions that do not promote any kind of positive living for the bacterium, an endospore can be made. Only the DNA is encapsulated in that endospore. So in our picture here, you have the dark center. I'm talking about the picture the furthest to the right. That dark center is the core. That would be the DNA material. It's encapsulated in the plasma membrane and the core wall, and then an outer wall that is made. That's this capsule we're talking about, and that then can live infinitely. I guess I mean I'm sure there are some that can't live that long, but some of them can live in because they're, quote, not alive. You can think of them as they're in suspended animation. And then when in fact the conditions become optimal, a new bacterium can be created out of the DNA. So the DNA can then code for proteins and the whole process can happen again. And sometimes this happens in terms of seasons, etc. And sometimes it happens because of intervention from humans that bacteria die off. Mobility and attachment are two things that are characteristics of prokaryotes. They have fimbriae, and fimbriae are different than flagella. We'll talk about all of these. These fimbriae, and if you look at the image on the left, the top left, the whole it looks like the whole bacterium. You have that longer flagellum. That one is used for taxis or mobility, for movement. And then you have the shorter ones that are more the attachment fimbriae. So the attachment fimbriae or attachment pillae. They use it. It is true that's what it's for, but that's not the same as a sex pilus. So they're two different things. That allows them to stick together and form colonies. It's one reason why they can form colonies. And then the idea of this in the upper left that looks kind of like a machine, it's an artist's rendition of how that flagellum is able to do a corkscrew motion. So it actually turns and whips. It turns and whips in terms of its movement. It's unique. It's unique with regard to its design, but it also helps make it be excellent for taxis, excellent for movement. With regard to the sex pilus, we have that in the bottom right. Only certain bacterium will have a sex pilus. So we do have what is considered in layman's terms a male and a female. That which has the sex pilus, so we think of it as the male or the genetic donor. That without is the female, and this is the way bacteria can exchange DNA, where you can have DNA changing or recombination with regard to bacterium. Some bacterium and most bacterial reproduction is by binary fission. So you have actually one cell turning into two, but you do need diversity, and there is diversity. And so you have this ability for DNA to go from one bacterium to another. And we'll talk about that in terms of the F factor and the R factor and how that helps create more diversity. And then in the bottom left, this doesn't come from your textbook, but I thought it was kind of interesting because we can all relate to E. coli. We hear about E. coli all the time. What you have is an artist's single E. coli rod, and you have these pili coming off, these attachment pili coming off, and what they have is adhesions at the end that can stick to protein markers on the out of a white blood cell, and in sticking to the protein marker on the out of a white blood cell, and then also sticking to protein markers on the lining of your intestinal lining, and they can just sit there, then colonize. And it was one of the reasons why E. coli makes an individual so sick and so painful, because you have mechanoreceptors and you have pain receptors inside your intestines, particularly your small intestines, and these E. coli can disrupt that. They can disrupt the environment of your small intestines, because once they attach, then they can replicate in your small intestines. And it does take some time and also some intervention, whether it be antibiotics or something, to actually kill them off. Now, your body will work on them. Don't get me wrong. That monocyte is a part of your immune system, but if your immune system becomes overwhelmed, there isn't anything that can be done. So that's a form of how those attachment pili work in your body. And then, as I said, with regard to motility, movement happens because of that long, very unique design of the flagella. The eukaryotic flagella is structured a little bit different and functions a little different than a eukaryotic flagella. The place that we find a eukaryotic flagella is in the male sperm cell, and you see one of those on the image to the left. That allows that sperm to have motility, to have movement up the in the female uterus, and then to the into the fallopian tube, so that the sperm can possibly meet up with an ovum. And then on the right, you have different designs with regard to flagellum. There can be a single. You can have several, or the bacteria can have several. They can have one on either end. And so that would be very interesting in terms of mobility. And then you can have many coming from all sides of the bacteria. So it is unique in its design to whatever particular bacteria you're talking about. And taxis is a way that a bacteria can either move toward food or away from a particular negative stimulus. So one of the things in terms of emerging properties is this ability to either move toward something that is beneficial to it or away from something that's potentially harmful. So movement tends to be a push forward in the evolutionary process. Other things that that are different is in terms of prokaryotic as opposed to eukaryotic. Remember, prokaryotic, no membrane bound organelles, and the DNA tends to be located in a single area, a nucleoid area. However, there are some membranes that have metabolic behavior, that have areas where just that particular characteristic is expressed. And in the aerobic prokaryotic that you have on the left, and this is a microscopic image of it, it shows you how small those are in angst units. Angst units, I'm pretty sure that's what the UM is. How small they are, and you have a specialized membrane. We're talking about that area that looks double layered, kind of looks like stripes on either of them, one for respiration and on the right for photosynthesis. These are adaptations to use oxygen or to use sunlight to make energy. We'll pick this up on the next one.